Hello, my name is Dr. Ray, and I am going to be doing the active learning session for you in class on the 3rd through 7 weeks of development. This movie I'm making for you so that you can watch the kind of like in a lecture style, the material for those individuals who like to sort of watch a lecture and watch a movie. And so that's what I'm trying to prepare for you. So when you come in to class, you can please do one of these 4 things. So if you already clicked on the link, then you're already watching the movie, but you can also complete the outline questions or read the chapter. So one of the things really should be done so that you can actively participate in class. Now I will say that I've been teaching for many years now, and I've also used my own videos to review material and remember how I presented certain material. And generally, you can watch my videos on 1.4 or 1.6. Speed is usually the speed that's appropriate for my voice and the amount of time that I pause between words. So in case you want to make the modification to that, go ahead and do that now. Here are your learning objectives. The learning objectives are, for me, and generally speaking for most people, they write them so that you know what the expectations are for you to complete that learning module. So you should be able to do these things basically by the time you finish learning from me and asking questions or doing some self-study at the end of the day before the exam. And for your own knowledge, you need to have this material in your brain somehow. So what we're going to be talking about today is the 3rd week of development, and then I'm going to kind of shuttle you through the rest of the week so that you can sort of see where you're going with this class. I know that embryology can be difficult. There are a lot of new terms and concepts, and if you have any questions, Dr. Hill and Dr. Korndorfer and myself are here to assist you. And so don't get too overwhelmed or discouraged that it's not something that you can tackle, because it certainly is. That's what we're here to help you with. And so essentially what I'm starting here where Dr. Hill left off at the end of 2nd week of development, where you had 2 layers in the embryonic disc, okay? And then we had a cavity here, the umbilical vesicle, he talked about that, and then we also had another cavity that formed on the dorsal side of this embryonic disc, although you don't know it's a dorsal side yet, but this is called the amnion. And so I need to get you to here, which is actually a 3 or a trilaminar embryonic disc. And so that's kind of the main event that happens in 3rd week. And the process that occurs that allows this to happen is called gastrulation. So this process involves a lot of cell shape changes and rearrangements of cells, movement of cells, and changes in the cell's properties and gene expression. The textbook goes into a lot of detail about those items, specifically which ones, and generally speaking, you don't need to know those or memorize those, except for in certain places where your other faculty may point something out that's super important, okay? But just generally keep the big picture in your brain at the end of the day. All right, and so during the 3rd week, this period of gastrulation is going to determine the axis of the body. So this is a dorsal view looking down on the embryonic disc, and we can see that something is happening here. It doesn't look uniform or homogeneous like the rest of the disc, that there is actually something occurring. And this actually is going to determine what side is right or left or up or down in this embryo, whereas before it was just sort of a blanket disc. And so it is a really important time. And although you can see that happening here through the primitive streak, which we'll talk about in 1 second, it actually had already been determined when Dr. Hill talked to you about the first 2 weeks of development. He discussed this area called the prechordal plate. And this is the area where we had these columnar cells that cuboidal cells that elongated into this columnar shape. These cells are going to have very unique properties. And this is actually when the axis was determined. But for the most part of being able to see something that shows you that the embryo is changing shape or position. That's going to be the presence of the primitive streak. And so I can't really stress enough that there are a lot of things that are happening behind the scenes in this story that we call embryology. And you know that you're not really talking about cells yet, but in your mind, please remember that we have a lot of cells that are moving around, and that these cells are influencing each other, and you have to have specific genes that can be changed that will affect these influences, and the abnormal expression of genes can cause major malformations in the embryo. And so this is actually really important, and although we don't stress it necessarily in these classes, it is playing a big role in malformations. And so this is just a quote that I really like, that was passed down to me by an old anatomist, that it's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. And it is true, everything has to happen just right to not have any kind of malformation occur in the developing embryo and fetus. So the first sign of gastrulation is the sign of this structure that we call the primitive streak. This primitive streak is a depression in the dorsal side of the embryo, which is an indication that cells are leaving this epiblastic layer, and it's forming mesenchyme inside the middle part of the embryo. So before, we just had these two layers here, the epiblast and the hypoblast, and so you can see them depicted here in the blue and the yellow. Now we're getting this other layer or space in between that is being formed by these migrating cells that are coming from the, really from the epiblastic cells, but it's causing this suppression that we call the primitive streak because it's visible. And then we have some anatomic parts of this primitive streak. The first one is called the primitive node. So it's just sort of the most cranial aspect of the primitive streak that we call this primitive node here. And the depression specifically we call the primitive pit. And then at the site of the node, as you move caudally, the rest of the whole depression together, that whole line that is a depression, is called the primitive groove. So these are just kind of anatomic points of the primitive streak that don't necessarily change anything, but when I'm talking about a particular area, it helps you kind of get an idea, because there are things that happen up here at the primitive node and more cranial to that, and so it just sort of helps you get that view. This is a transverse view cut down through the embryo, and so here you have the umbilical vesicle here, and up here would be the amnion. So I've cut the embryonic disc in a transverse view. This is a superior view, so I'm looking down as if I were in the amniotic cavity. I'm looking down on the dorsal side of the embryo. Okay, and so this primitive streak, then these migrating cells, actually add length to the caudal end of the embryo. So they start here, and it makes the embryo longer. And this is, again, a dorsal view looking down on the embryonic disc. So this is the part that was added through the process of gastrulation. So hold on to the big picture here. The big picture is that we started off with this bilaminar embryonic disc that had an umbilical vesicle and an amniotic cavity. Here you are looking at a sagittal section that's cut through this disc. Now, if we take this disc and cut it this way and turn it, we can see it in a transverse view. And I kind of want to just take a second to point this out, because you're going to be using pictures to study embryology a lot, because it's not something you can see in the lab, that you can't see these developmental processes occurring. And I want you to pay attention to the actual cut of the embryo, and that will help you kind of keep things in a line. And I know a lot of students even kind of draw them sometimes, and that kind of helps, because it's not difficult, it's just hard to picture. And so when you look at a picture, don't just look at it and look at the colors and just look at the lines and what the labels are. Try to think about, what plane is this? And that will help you put it together. And so that's kind of what this slide here is for me to remind you to do that. And so here we started off 2-layer, we have these cells, and now we have 3 layers. And that brings me to another topic that we have to discuss briefly, or otherwise it just starts to get all jumbled up, is what is a mesenchymal cell? So I talked about the epiblastic cells moving through gastrulation and becoming mesenchymal. So they undergo a transition. And this transition is from the kind of undifferentiated cell before into a cell that's called mesenchyme, or a mesenchymal cell. It has unique features. So I kind of highlighted the most important ones. They're very loosely organized. I know you're not looking at cells yet, but when you do later, you can kind of come back to this and see that these cells are not tightly bound together like the other cells that they show you in pictures. I mean, even if I went back to this one, you can see like they're drawing cells very close to each other for a reason. It's not because it's just sort of someone just came up with it. It's because these cells are close together. They're held together by junctions. But here, these cells are loosely organized. They can move around. They can phagocytose things, and they're pluripotent, so they can become so many different types of tissues. And generally, all of embryonic connective tissue is going to come from mesenchyme. And I actually added in here an article for you if you want to look at this further, and it's talking about mesenchyme and regenerative medicine. So if you're interested, mesenchymal cells are very special cells. Okay, and so what are the fates of these migrating mesenchymal cells? All right, so they came through the primitive streak from this epiblastic layer, from this primitive streak, and they're moving somewhere. So some of the cells become intraembryonic mesoderm. So now we're going to end up having this new layer, the middle layer, and this layer is called mesoderm. Please, it's very difficult. I know the words all kind of sound similar. Mesoderm is not the same as mesenchyme. Mesenchyme is that special kind of cell that I just talked about before that has unique properties and it has unique fates. Mesoderm has its own fates. It's going to become its own thing. Some of those fates cross over because all of these cells that created the mesoderm are mesenchymal cells. But mesenchyme, there are other layers that will also go through a mesenchymal transition to become mesenchymal cells, and so they're not synonymous terms. So some of these cells will become intraembryonic mesoderm. So this is mesoderm that's inside the embryonic disc. That's what intraembryonic means. And if there's an intraembryonic mesoderm, you better believe that there is an extraembryonic one. But right this second, let's focus on the intraembryonic mesoderm. Okay, some of the cells through the primitive streak are going to replace the endoderm, the hypoblastic layer, and this is going to become what we call now the endodermal layer. So endodermal layer is going to come from these mesenchymal cells. There were cells there before, and that's what this artistic representation is showing you, that these mesenchymal cells are going to completely replace those previous cells. And then the remaining cells in the epiblastic layer, we're going to call now the ectoderm. Okay, so we're going to end up with 3 layers now: the endoderm, the mesoderm, and the ectoderm. All right, at the same time that the primitive streak is doing all this really fun stuff, there is also another thing that's occurring, and that's the formation of a process called the notochord. Okay, so we don't only have these cells migrating. This is a dorsal view, by the way. We don't only have these cells migrating and creating growth at the caudal end of the embryo. Some of these cells are also migrating cranially, and is causing expansion in a middle layer in the cranial aspect of the embryo. Okay, and some of the cells in the midline, these mesenchymal cells, are specifically going to develop into a special structure called the notochord, or the notochordal process. So this is made from mesoderm. I just messed up. I apologize. Mesenchymal cells that are in the midline of the embryo. The midline of the embryo's mesenchymal cells that have migrated are going to make a process called the notochord. So if I cut this embryo in half and I showed it this way, I'm looking at the embryo in a sagittal view here, okay? And so this process is going to develop in cranial to this primitive pit and primitive node, all the way cranial to the precordial plate that we had. Do you remember the precordial plate being that site at the cranial end of the membrane where we had the special cells that sort of started this whole business? The precordial plate is not going to receive any migrating cells from the primitive streak, and it's going to remain a fused layer of 2 layers from where kind of the epiblastic cells and the hypoblastic cells were, but we're still going to call them now the ectoderm and the endoderm because we've already transitioned this embryo to 3 layers. So this site, this precordial plate, is going to remain 2-layer, and it's going to be the site of where the oropharyngeal membrane is going to be located at. And that will become important anatomically later. All right, and then we have another area where we will not receive any migrating cells from the primitive streak, and that is at the caudal end of the developing embryo. And so very similar, you're going to have ectoderm that's going to remain fused to the endoderm. And this is going to be the area of the cloacal membrane, okay? And so this is eventually where the anus is going to be, the oropharyngeal is where the mouth is going to be. So these are 2 important areas. Okay, so the pit that we had, remember the node that had the depression we called the primitive pit? So the primitive pit, now that we have this special area that we know is going to develop into a nodal cord, right now it's just cells, it's going to expand, and it's going to sort of run down the center of this area that the developing nodal cord is in. And we're going to call this depression that expands the notochordal canal. So I have a little, if I click, you can kind of see it expand longer. And so it's pushing these cells out and making this canal in the middle of where these mesenchymal cells were located at. And so there it goes, and it's going to become even longer. So now we can see this notochordal canal that has developed. And so what's going to happen after that is that the floor of what this notochordal process that's developing is going to actually start to degenerate and break down. So I have a little animation. You can see it's starting to develop holes. The holes are going to coalesce with each other, and they're going to disappear. And now we're going to have the floor of this developing notochordal process just sort of breaks down, and we have open communication with the amnion and the umbilical vesicle. So this is only happening in the midline of the embryo where this notochordal process is developing. Okay. Now, the proximal part of this canal is going to kind of persist longer than the whole rest of it, and it's thought that this helps equalize pressure between these 2 fluid-filled cavities. And so we call kind of that the neuroenteric canal. You totally do not need to know that. I'm just trying to help you complete the story, because when you don't fill in the pieces, it becomes more confusing than me just sort of explaining to you how pressure stays equalized through this amazing process. Okay, so in this slide here, I was in a sagittal section, so this was like the head region of the embryo. Here's the tail region. And then, I'm sorry, I totally said that backwards. This is the caudal region. This is the cranial region. This is moving upwards. And I am going to switch views now. So a cut in a sagittal section, but I'm going to switch now to a transverse view. So now we're looking at a transverse view. So like these are the lateral aspects. This is the midline or the medial aspect of the developing embryonic plate. Here you can see the notochordal process developing these cells that are going to become part of this process, and the floor that has degenerated. You can't see the hole that goes between the 2, but that's okay. We're at a different plane of section. Okay, so this notochordal process is going to involute. So you can see it sort of, the edges are going to come together, and they're going to come into close approximation with each other, and they're going to fuse, and the endodermal layer is going to be restored. That's cool, huh? Now we still have the endoderm. We needed that. It has to be a continuous layer because it's going to develop into the gut. And now we have this structure, this special structure in the midline that's called the notochord. And we're going to pick up on what that becomes later on, okay? And so now everything is as we need it to be, because this is going to have its own fate. Now we have mesoderm that had migrated from the prismatic street on lateral to both sides of this midline structure called the notochord. So, in summary, by the middle of the third week, so through the development, we are going to have mesoderm, this new layer, and everywhere in the embryonic disc except for 3 places. At the oropharyngeal membrane, cranially, that will become the mouth. That layer only has ectoderm and endoderm. At the cloacal membrane caudally, that's where the anus will be, we only have ectoderm, endoderm. And in the middle of the embryonic disc, in the median plane, where the notochordal process developed at, because that has a special structure that we call the notochord. Okay? That's just in summary. Now we're going to fly through a little bit of what happens just to get you going, because we're going to pick on this up. Your class, your other faculty and myself will help pick this up throughout the rest of the course. Neurulation. Neurulation is a special process that is going to be a result of the notochord developing. It's going to induce the cells that are dorsal to it to develop into the neural tube. So I'm going to talk to you about neurulation next, and you can see how that happens. It's pretty cool. And so this is to be continued, okay? Now, the mesoderm that is going to be on the lateral aspects of the notochord is here, depicted here, is going to further differentiate into 3 main regions. And these things are going to develop into very specific parts of the body. And that's why it's kind of nice to know kind of figure that out now. And so I like this picture. At first I really didn't when I was first learning it, but now, because I don't think it was sort of explained thoroughly, but I color-coded it for you. So the paraaxial mesoderm is one of the layers, and that is going to be the most medial mesoderm that's just on each side of the notochordal process. The purple area is the area that we call intermediate mesoderm, and then the green area is called lateral mesoderm. And these are the 3 differentiations of the intra-embryonic mesoderm. And that only becomes super important right now and as you take a step back and think of the embryo as a whole, because you can see that these mesodermal layers are continuous with the mesoderm that is extra-embryonic or outside of the embryo. And we will pick this up and how this is sort of this nice symphony of occurrences when we talk about development of the fetal membranes. Okay, so paraaxial mesoderm very specifically is going to develop into these interesting blocks of mesoderm that we call somites. And they are going to give rise to the axial skeleton, and all of the musculature and dermis of the skin and everything is going to come from paraaxial mesoderm. And the first one to develop is called the otic placode. The otic placode, which is going to be in the region of the developing ear. And it's not very hard to appreciate, or I think maybe now it's very easy to appreciate, that the nerves that are going to develop in these somites, see how close they are to the developing spinal cord? They're right nearby. And so although everything grows peripherally and goes outward, they start really close to each other. And so it kind of helps to put this picture together later. It's how is it that something that ends up being way out here has a nerve and they don't grow out like through the tissue. They actually started and everything grows. And so that sort of is a little bit helpful to kind of appreciate this now. And so I just wanted to give you, this is the interesting view of the dorsal side of an embryo, and you can see what those somites look like. They kind of look like blocked ridges. And they develop really fast. So they have, at 22 days, you have 5 of them, and then they're growing, then you have 13, and you get all the way up to 35. So we're going to continue what those turn into later on. The next layer of mesoderm, so here you have paraxial, and here you have the next one. It's a smaller section of it, and we call it intermediate mesoderm. And the intermediate mesoderm will give rise to the entire urogenital tract. So just if you're trying to place that somewhere, it is to be continued. We will follow that up later on in the course. And then the lateral mesoderm is kind of a larger section, And it's going to develop into two regions because spaces are going to develop that will divide lateral mesoderm into two spaces, okay? And it's going to divide it into two regions that will be functionally different from each other. It's very important. So what this space that occurs doesn't just sort of haphazardly occur. This space is occurring because we have the development of a space inside the embryo called the intraembryonic coelom. And so you can see that it is going to run sort of all in this area. It's a horseshoe-shaped space that develops here. If I cut the embryo in a sagittal plane, though, I can only see a little tiny part of it where it was cut here. If I look at the embryo at this plane of section, I can see that it has wide communication with the extraembryonic coelom, or the extraembryonic spaces at this time. So this is sort of a space. It's not just transitional, though, because they will develop into structures that will remain in the fetus and throughout the adult life. So, for example, the intraembryonic coelom, this wide space, and this is much farther along. I just want to show you that it's going to eventually develop into body cavities. So the pericardial cavity, the pleural cavities, and the peritoneal cavities will come from this intraembryonic coelom. And here you can kind of see the artistic rendition that is communicating with the extraembryonic coelom at the time. Okay, so I talked about the lateral mesoderm dividing, and I don't want this to be a very small point in your in this, and hopefully you're not glossing over here with all of the details that I'm throwing at you. The great thing is you can watch the parts that are hard multiple times, and we're going to talk about this in class. But the lateral mesoderm is going to separate into two parts, and this is just looking at the pink part, just the mesoderm. The part mesodermal layer that's connected or adjacent to the overlying ectoderm is called somatic mesoderm, okay? And the mesoderm that is going to be adjacent to the endoderm is called splanchnic mesoderm. And actually, we usually talk about them together. So the somatic mesoderm with the overlying ectoderm we call somatopleura. And the splanchnic mesoderm that's connected to the endoderm we talk about together as splanchnopleural. And these are going to be functionally separated when we talk about the nervous system being separated into GVA and GSA. So we have somatic innervation and we have visceral innervation, and this is where all of that comes from, is from this division of mesoderm. Okay, and so we will continue this. You've already been introduced to the nervous system, I think, by now, and this will be something that you kind of continue to reinforce in your mind throughout your training. I want to talk very briefly about a structure that shows up in all of the images and people talk about occasionally, but never doesn't have a huge role, and so it kind of gets glossed over to the degree that you just want to know what this is. And so there is a small diverticulum in the umbilical vesicle that begins in the 3rd week, and we call this the allantois. And it extends up into the connecting stalk. We're going to continue with this when we talk about placental membranes and development of the placenta. And later on, it's actually going to be continuous with development of the bladder. So the allantois doesn't have a very big role, but it does persist throughout most of development. So you're going to see it a lot in the pictures. And later on, we're going to call the region that persists the urachus. And although it doesn't have a huge role, that the urachus and allantois doesn't play a huge role, the mesoderm around it does. So the mesoderm around this region is going to develop into the umbilical arteries and is also going to be one of the first places to produce blood cells. So it is something that is important. Speaking of blood cells, the cardiovascular system is the first system to develop. And at the beginning of the 3rd week, we start to have some blood vessels form in the umbilical vesicle. And so we're real zoomed out here. You can't see the embryonic disc depicted here because it's hiding in here. But we're looking at the wall of the umbilical vesicle, and you can see it kind of looks bubbly. The artist is showing you that we have these blood islands that are developing blood cells, and it's also developing in the connecting stalk as well. And we'll talk about the chorion later, so don't get bogged down with that terminology. Okay, and so blood vessels will also start to develop after these blood islands develop in this extra embryonic mesoderm. And so this is really important because we don't have a vascular system that's developed in the embryo yet. We're just starting this off. So it's not actually going to start inside the embryo. It's going to start extra embryonically. And so there are two processes that occur for vascular system development, and this is something that you just need to know for being in medicine. The first one is called vasculogenesis, and that's the one that we're talking about right now. So forming new vascular structures by assembling the angioblasts. These are special cells that come from mesoderm. And so they come from mesoderm, they only come from mesoderm, and they are going to assemble into these blood islands and form vessels. Angiogenesis, however, is for the formation of new vessels by the budding of pre-existing vessels. This can occur and will occur in the body throughout life, and it is an important process of the repair process. And so this is something that you need to kind of hold on to. So vasculogenesis has to be the first step. So we have to make a blood vessel before we can bud from new vessels. Okay, and so what happens is these angioblasts that come from mesoderm will aggregate and form clusters. We call them the blood islands. We just saw some examples in that cartoon picture a few slides back. And then these small cavities will form in these blood islands. Okay, some of the angioblasts will flatten out, and they will become the lining cells of the blood vessels. And we call those lining cells endothelial cells. You will become best friends with endothelial cells. Then what happens is that these developing vessels fuse with nearby developing vessels, and they make larger vessels. And so, and then they're going to bud off through the process of angiogenesis. So that's how this sort of all gets started. And so the part that I want to reinforce to you, this is another tripping point, something that's easy to get confused, is that endothelial cells, the lining cells of blood vessels, come from mesoderm. It's really easy to get them confused with the endodermal cells, endoderm, embryonic endoderm layer. That's not going to become the endothelial cells. Endothelial comes from mesoderm. Endodermal cells, they're going to develop into the gut. Okay. All right. And so I just want to point out here that the blood cell development that happens intra-embryonically, so within the embryo, doesn't occur until the 5th week. So that will be picked up later when you do the other cardiovascular development. So most of the processes early on, they're all going to come extra-embryonically, although you start to make blood vessels. All right, so the cardiovascular system is the first to develop. So if you remember, some of those cells, those mesenchymal cells coming from the primitive streak, moved cranially and are going to come together in a special region that we are going to see the heart develop into. We call this area, which it is mesoderm, so it's between the ectoderm and endoderm, we call this area the cardiogenic mesoderm. So that's going to be sort of this area where the heart will develop. Now, it's not going to be way up here when we'll talk about folding of the embryo and how it moves around, but it is going to be an important spot. All right, and so you're going to continue the development of this cardiogenic mesoderm and the structures around it when you do heart development with Dr. Hill. All right, so we're kind of to the last part, yay, and that is how do we get this embryo that is a flat disc to look like a cylinder, an actual embryonic shape, because a lot of things are going to have to happen for that to occur, and I get to explain that to you, yay. So firstly, the embryo is going to fold in a median plane, and so that means I've cut this embryo in a sagittal section. So this is the head region, this is the tail region, or cranial and caudal. And okay, so as the head folds, in the head region, the developing brain is going to get, it's going to really push the embryo this way. So it's going to grow faster than the rest of the embryo. And as it pushes the embryo forwards or cranially, it's going to curl ventrally. And it's going to tuck everything that was here, that was more cranial even than the developing brain. It's going to pull it on the ventral side of the embryo, and it's going to allow the brain to overgrow, or it's going to overgrow all of these structures. Okay, so we had this cardiogenic area where we're going to have development of the heart. So that was actually cranial to the developing neural tube, which I know you haven't talked about the neural tube, but just this is the brain. And so as that pushes and folds ventrally, all of these structures, the heart and this structure called the septum transversum, which is sort of like a partition, is all going to be pulled ventrally. And another side effect of this, or something that's going to happen, is that the endoderm that was in this region gets incorporated inside the embryo. And we're going to call that region the foregut. And that's going to be important in further development and also in anatomy and lots of different places. Okay. So after it's folded, the septum transversum, which you see here, this is going to be the partition between the thoracic and the abdominal cavity, and that will be carried on and continued later on in other lectures and places. But you can see that the pericardial cavity and the cardiac region is going to now be on the ventral side of the embryo. The brain and developing spinal cord neural tube is going to be completely on the dorsal side at this point, and all resultant of the head fold. Now, on the other side of the tail fold, a similar thing happens. The spinal cord is growing, and neural tube is growing faster than the rest of the embryo, and it's going to push the structures that were out here onto the ventral side of the embryo. And so you can see what those structures are. The connecting stalk, that famous atlantois I just talked about. So this is where the umbilical cord is going to be. It's not sticking down between the developing embryo and fetus's legs. It's going to be on the belly side, right, on the ventral side. Well, that happens because the tail folds under and the umbilical cord or the developing connecting stalk gets pulled onto the ventral side of the developing embryo. And another side effect is that the endoderm that is in this region, the endoderm, is going to get incorporated inside the embryo as the hindgut. Okay, so that's going on here. And so this area, if you remember, the cloacal membrane is going to be the future side of the anus. It also gets pulled instead of being on the back side, the dorsal side of the embryo, it's going to get pulled down in this region more ventrally than it was. Okay. And so this hindgut is going to develop into the urinary bladder and rectum. Does get incorporated as the hindgut, but this area here, the cloaca, is the part that's going to develop into the rectum and the bladder. And it's going to expand and dilate, and Dr. Hill will go over that when he talks to you more about the development of the urogenital system and the digestive system. Okay, and so that is a resultant of the tail folding, the umbilical cord being on the ventral side of the embryo. Now, it doesn't only fold in the median plane. The embryo also folds in the horizontal plane. And so you can see here, this is a new view of the embryo. I'm actually cutting the embryo transversely and looking down on it. Let's pretend like we're looking down on it. So here you can see where the extraembryonic coelom is communicating with the intraembryonic coelom. You can see this is the area the amnion would be. This is where the umbilical vesicle is. So this area here is going to move ventrally and is going to come together. So these sides fold down in this horizontal plane, and they meet in the ventral side of the embryo. Now this one's a little bit harder to picture. I think the head and tail folds is a little bit easier to grasp from a diagram. This one's a little rougher. And you also have that separation of mesoderm, the somatopleural here and the splanchnopleural. Those things are coming around. So the somatopleural, the body wall, is coming around and coming ventrally to meet the other side. Now, the umbilical vesicle is still sticking out here for quite a while in this area, connected with the gut here, the endodermal layer, and the connection will be the midgut. And so we'll talk about, Dr. Hill will talk about development of the midgut and development of the gut in entirety, and all of this will make a lot more sense. And so at first, if you remember, the umbilical vesicle freely is connected to and it communicates with the endoderm. And then as it folds, though, the communication gets tightened down and much more narrow. And we call this little communication until it disappears the omphaloenteric duct. And that you'll see that in a lot of the diagrams, but they don't really talk too much about what it is. So don't get too confused. It's not this really big new thing. It's really just a communication. Okay. Now, the other really cool thing about this folding in the horizontal plane is here you had the amnion on the dorsal side of the embryo before it folded. But how many people, if you've seen a baby delivered, you know that the amnion is all the way around the baby. How does it get all the way around the baby if it just starts here on the dorsal side? Well, it's because as these folds, they come down and come together, this is all the amnion. The amnion gets wrapped all the way around and is connected to the outer portion of what is, I know they're showing you the umbilical vesicle, but let's pretend like it's in the spot of the umbilical cord or the connecting stalk. So that's pretty cool. And I'm going to talk to you about that further when we do placental membranes. Now, this is a link for you if you want to look at an animation of the folding of the embryo. I don't really talk through it. It's just sort of like labels and stuff, but I still think it helps to see how it happens. You can watch it 100 times if you want to sort of get the picture. And I just want to briefly talk about how weeks 1 through 8 is considered the period of organogenesis, okay? And that is this period here, and you can see how all of these systems are developing. All of these systems it's the most important, weeks 3 through 8 is most important for developing of the organ systems. So if you have any kind of disruption from teratogens, then you're going to have malformation of these systems. And hopefully you're starting to hold on to, at the end of the 8th week, this no longer an embryo, the developing embryo transitions and is now a fetus. Okay, so. And so that's why these weeks 3 through 8 are so important, is because you have these systems developing, and that's why the embryo looks the way that it does. So because of these developing systems, it looks unusual. You have somites, here you have pharyngeal arches, here you can see the heart's huge, and you have this brain, you have these eyes and ears that are developing. And then by the end of the 8th week, the embryo is now a fetus. This is the end of the embryonic stage, and it has a very distinct human appearance. Now, what you need to know is which structures are derived from which germ layers. And when I first learned this material, I sat down with flashcards, and I made flashcards for everything so that I would know that if it was a pigment cell, it came from which layer. And that really helped me answer one question, and that was the spleen, because the spleen doesn't come from what you think it would, the endoderm. The spleen comes from mesoderm. So I did get one question right because of it, but it was a lot of wasted time. What I want you to do right now is start to think of really just some really important ones that you want to hold on to. Like ectoderm, I highlighted the ones that are kind of most important that you start to think of. That ectoderm is going to develop into the nervous system. When we talk about the neural tube development, you'll see why. It's going to do all the epidermis. You're going to do all of the appendages that are attached to the epidermis, so hair and nails. And it will fill in all of the other little ones as the systems are unveiled to you throughout this course. And so neural crest cells, because of the development of the neural tube, come from ectoderm. And other things too. Pretty much because of the neural crest cells coming from ectoderm, anything in the head region can come from ectoderm, because neural crest cells develop into so many different things in the head region. And then when you think about mesoderm, I want you to already start to think about some key things. So a lot of the connective tissue of the embryo comes from mesoderm. Uh, um, muscles come from mesoderm, whether they be striated or smooth or cardiac. All of the blood vessels, and at the beginning, all of the blood are going to come from mesoderm. And then some other things, because of the intermediate mesoderm, you're going to have the whole urogenital system that is going to come from mesoderm. And when you think of endoderm, I want you to think about some big players here. I want you to think gut mainly, but also the lining of the respiratory tract too, also comes from endoderm. Now the surrounding connective tissues and muscular layers are going to come from mesoderm, but the linings that are made up of special cells called epithelial cells are all going to come from endoderm. And then other important digestive organs like the liver and pancreas are going to come from endoderm, and then the actual bladder and urethra, the lining of these structures are going to come from endoderm, not the muscle. Okay, so hopefully I gave you a good introduction. There are a lot of things going on between weeks 3 and 4, and mainly, and then I kind of just showed you a little bit of where we were going with the other weeks to help you get started. I want to thank one of my mentors, Dr. Whitworth, for his help. I always do, because he taught me and gave me a lot of his presentations to work off of. If you have any questions or comments, please feel free to email me. I also have a Facebook group if you want to join. I have a lot of histology on there, but I'm going to be adding development and growth to it as well. And I'm also getting started with Instagram. So thank you for watching. I will see you in class. I hope that this has been helpful.